So while I'm loading the slides, for anybody who doesn't know Celldom yet, we are a small company located in California, in San Francisco Bay Area, and in San Diego. And we are manufacturing and developing tools for live cell assays. So, and I also want to thank Alex and the Selector team for inviting me to present our technology here and some of the workflows that we are developing together with Selector. I'll start my talk with a short introduction of the Celldom platform and some of its capabilities, and then go through 4 applications that are relevant for immunology. The Celldom Live Assay platform consists of 3 main components: the clone express plates, which are practically a micro well array bonded onto a 96-well frame. Then we also developed a live imager called the Clone Explorer, which has an integrated cell picker that can isolate cells from user-selected microwells. And we are developing software both to run the instrument and to analyze the images and data visualization. These cartoons highlights some aspects of our well plates. So from the outside, as Alex showed, they look like conventional 96 well plates, but if you zoom into a well, you can find approximately 4,000 microwells in each well, so almost 400,000 microwells on a plate. The side view highlights some unique features of our microwells. They are conically shaped, they are very deep, and the rims between the microwells are very thin. So, and that prevents cells from getting trapped on top of the rims when loading them. Priming submerges the microwell array in culture medium, and then it's ready for cell loading, which is extremely simple and efficient. We just need to pipette cells on top of the microwells. They will sediment by gravity, and all cells that have been loaded will end up in microwells. There's no dead volume here. One thing that I want to point out here is that all the 4,000 microwells in a well have access to the same volume, and soluble molecules can diffuse in and out of the microwells. However, the cells, once they are seeded, especially suspension cells, they stay in place, and we can replace partially the media on top of it to feed cells, and also to add and remove labeling reagents. The plate loaded with cells can then be inserted in the imager. On the left side are photographs with the door closed and door open. On the right side, a cartoon. So the plate is inserted in the incubator that is temperature controlled, humidity controlled, and also CO2 controlled. The microscope can image any time during culture in bright field and 4 fluorescent channels. And the cell picker consists of a syringe mounted on a XYZ stage with a very thin stainless steel needle that can access individual microwells, retrieve cells, and then deposit them in a deposition plate before being washed and prepared for the next step. So the whole process is automated. Our image analysis pipeline starts with identifying and indexing all microwells. In the second step, cells within each microwell are segmented, and certain characteristics for each microwell are generated and saved, such as the cell number, fluorescence intensity, etc. In this graph, the colored rectangles signify different cell numbers per microwell. So yellow is microwells with 1 cell, magenta 2 cells, blue no cell, and this can be used for calculating cell distributions after loading. And if you see here on the blue line is from a condition where 4,000 cells were loaded per well, the same number as microwells, and they follow roughly a Poisson distribution, where we get 1/3 of the microwells empty, 1/3 with 1 cell, and 1/3 with 2 or more cells. If you want to have higher occupancy rates, you need to load more cells, and the distribution shifts to the left, we get fewer empty microvels and more with 2 or more cells. Up here is shown a zoomed-in image of 1 microvel over time that was loaded with 1 cell that divides 3 times. Those cell counts can then be used to calculate growth rates for each microvel, and it's plotted here for across the plate for each well. And as you can see, we get very uniform growth conditions across the entire well. Another aspect that I want to point at that also on average it's very uniform, but we can also identify very rare outliers such as fast-growing cells. This slide shows the capability to recover the cells from individual microvels. For this experiment, we did the mixing experiments. We first mixed 2 cell lines that are differently labeled, one GFP labeled, one mCherry labeled. We mixed them together and seeded them at low density and incubated them while imaging. And then we attempted to demix those 2 green and red cell lines by isolating monoclonal cell lines, which is shown with this gating strategy here. So we selected cells, microvels that were seeded with a single cell that were either mCherry positive or GFP positive and were fast growing. After gating, this generates a list of selected microvels that also contains the coordinates of the microvels that can then be read out by the Clone Explorer machine to guide the tip into the selected microvels to recover the cells, then deposit them, we cool them up, and confirmed in secondary assays that we indeed selected and isolated monoclonal, exclusively green or red cell lines. We also have image documentation during each step of this process, which I'll show in a video in a moment. So the video starts with the microvel array after loading, and will focus on one microvel that has been loaded with a single cell that's mCherry positive, as shown in the Texas red channel here. So microvel with 1 cell, that's this microvel. So over time, the cells divide, they stay mCherry positive and GFP negative. Now, before we pick the cell, we take images to confirm that indeed it's GFP negative, and mCherry positive. And now the picking needle comes in, and the computer software guides the needle into the target microvel. It identifies. Yeah. So what's to save time, I'll just explain that the microvel was then guided in the target microvel, removed all the cells, and we take an image afterwards showing that the target microvel is empty and all the surrounding microvels are not disturbed. Another capability of our system is a secretion assay, where we can measure secreted molecules secreted by single cells. Alex already described most of it. So briefly to recap, we code a capture antibody on the surface of our wells, then seed cells, in this case T cells with and without antigen-presenting cells, and then culture them over time. And during this culture time period, we add a fluorescently labeled detection antibody that then accumulates where interferon gamma is secreted, and we can detect interferon gamma secretion in real time while it accumulates. Interestingly, we found that the signal we found a very strong signal around the perimeter of the microvels, which builds these very characteristic secretion rings, and we trained an algorithm that automatically detects interferon gamma positive microvels. To establish this assay, we used the mouse OT1 model, which consists of OT1 T cells that are with 1 specificity for an antigenic peptide within ovalbumin. We tested 12 different conditions in a plate here. In columns and rows are 8 replicates. Each square represents a small bar chart of number of secretors per well on the y-axis versus 24 hours of time period. And I want to draw your attention to just column 5 and 6, which contains the OT1 T cells with unpulsed dendritic cells. There's no background signal. And when we combine them with pulsed dendritic cells, we get a rapid and strong secretion signal. And now this brings us to our first application, which is a no-wash kinetic fluorospot assay, which offers a few advantages over conventional ELISpot assays. The workflow diagram is shown for frozen PBMCs, the starting materials that are thawed on the day before assay, arrested overnight, then counted, seeded in the plate. Then we add stimulation peptides. For development of the assay, I used a mixture of viral peptides from CMV, EBV, and flu called CEF. And we also add the detection antibody, as described previously, at this time. And then we just need to place the plate into the imager. Imaging and analysis are automated. So this shows the results of a full plate run with PBMCs from one donor. We tested 4 different media, and we seeded cells at 2 different cell densities. We used rather high cell densities as we wanted to detect rare events, so 100,000 or 200,000 cells per well. We included a lot of negative control wells, so DMSO as stimulation reagents, as we had expected that we would see quite some variability in background, but somehow for this donor it was completely clean, no secretion at all. And when the cells were stimulated with the peptides, we see rather strong secretion and a roughly 2-fold increase in the number of interferon gamma positive microvels when we double this cell dose. We also see an effect of cell culture media on the number of secreting cells. I want to briefly summarize the highlights of this assay. It has minimal hands-on time. We get a kinetic readout. Both analysis and detection are standardized. And as an additional bonus, the cells can be isolated from selected microvels after the assay. The next application is the assay that Alex already described. I'll briefly show some results. So we did a brief proof of concept study. And the question behind this experiment is, can we identify the specificity of a TCR of interest? So in this case, we tested with a CMV specific TCR that Selecta cloned into their Chukat reporter system. And they also provided the K562 antigen-presenting cell library, which expresses 100 different peptides at roughly equal frequency. So that the CMV peptide was approximately 1% of the cells expressed the CMV peptide. So the unique challenge of this assay setup is we are interested in the information of the peptide, which is encoded in the K562 cell, but the readout comes from the Chukat cell. So you cannot sort activated Chukat cells because then you're missing the K562 cells, which makes it very uniquely applicable to our microvel system, where we loaded 1 Chukat cells and 1 K562 in individual microvels, then cultured them and monitored activation. We then isolated microvels where we saw a GFP signal, and those were then sequenced by Selecta, and most of the recovered sequences were indeed from the CMV peptide. Yes, we still have time. The next application is discovery and validation workflow for TCRs in as little as 1 month, where we use the dendritic cells as antigen-presenting cells that are derived from PBMCs. While we generate those, we also enrich antigen-specific T cells by co-culture with the antigenic peptides. We then combine the mature dendritic cells that have been pulsed with the same peptides and the enriched T cells in the microvels, monitor interferon gamma expression, isolate hits, then expand the hits, the cells. And once we have sufficient numbers of cells, about 2 to 3 weeks, for validation, We have multiple choices depending on if there's a tetramer available. We can do tetramer staining to validate the hits. We can retest them on our platform with antigen-presenting cells. And for this, we like to use the selectors HLA expressing K562 cells. And we also will freeze a portion of the expanded cells for TCR sequencing. I want to also mention that this workflow is very flexible. We have tested several different antigen-presenting cells for both discovery and validation. And we also have successfully deposited the hits in lysis buffer and then determined the TCR by direct sequencing of the exported cells. And I'll show you some results from this workflow. That was the experiment was done together in collaboration with Selector. This is a FACSTAR. So then we use the MART1 melanoma antigen as a model system and donor PBMCs. After the 1 week pre-enrichment, we see about 100-fold enrichment of T cells specific for MAT1 by flow cytometry. And those were then seeded on our plates together with pulsed dendritic cells. Here are the gamma secretion data. All the negative controls on the right side had none, no, or very low number of secreting microvels, versus the positive control, which was consisted of anti-CD3, CD28 antibodies, had hundreds of interferon gamma positive microvels per vels, and our experimental condition T cells with MAT1 pulsed dendritic cells yielded between 10 and 30 hits per well. We then isolated those positive cells based on following gating, so interferon gamma secreting, growing cells that was also CD8 because MAT1 is a known CD8 antigen. The pictures here on the top right represent from a representative microvell that was seeded with 1 T cell. After that, with 2 dendritic cells on top of that, we see increasing interferon gamma signal over time, and after about 5 days of culture, this microvell was full with T cells, with cells that were positive for CD8. We isolated that microvell, and here's a picture of the same microvell after picking, which is empty, expanded it, and this clone was then virtually 100% MAT1 positive by tetramer stain. We isolated several cell lines, and 10 of them were sequenced by Selector with the AIR TCR-seq, and they identified 7 unique TCRs, 3 of which were found in 2 cell lines. So TCR number 1 was found in 2 cell lines, 2 and 4 as well. In this table, there are also the CDR3 sequences listed for alpha and beta chain. While sequencing was underway, we also revalidated those cell lines on a Celldom platform, and for this we used the selectors, the K562 expressing HLA, and we titrated the MAT1 peptide to derive dose response curve, so dependency of peptide concentration and numbers of secretors of wells, and we used these to calculate EC50 values for the different TCRs. We found that the EC50 values for pairs of cell lines that express the identical TCRs were very similar. So the first 2, approximately 1.5 nanomolar. And in addition, we also found that we identified TCRs with different EC50, with different affinity. So the highest one in this system had 5 nanomolar and the lowest 0.1 nanomolar approximately. So this is the last application, which I quickly go through, CAR-T potency assay that we just completed a proof of concept with CD19 CAR-T cells and GFP expressing NALM6 as target cells, where the readout is cell killing of NALM6 visualized by disappearance of GFP. We also detect interferon gamma secretion, which will show up in the Texas word channel here. And we stained the CAR construct after killing. So 48 hours after start of the assay, we added the CAR detection reagent, which is an anti-idiotype antibody against the CD19 CAR, which also will show up in this channel. Yes, so the one more note to this video, that the time frames are very different along the video. So initially we imaged every 30 minutes, then every 4 hours, and then the last time point was 4 days later, just to show that the cells expanded, and we'll see a full microvell at the end. So we started with, after the first images after loading T cells, and this microvell was loaded with 1 T cell. In the next frame, there are 3 NARM6 GFP added, and the cells interact with each other, and slowly the GFP signal that cells are killed versus the interferon gamma signal, and then this was the CAR signal. Maybe one more time. So the interferon gamma signal starts to appear, and then we'll see the CAR stained cell, and at the end, the full microvell. We also did some further analysis of specific wells. Here's two graphs. So we tested 2 different batches of T cells, and we looked at effector to target ratios 1:3, which is 1 T cell and 3 NARM6 versus no T cell and 3 NARM6. So the NARM6 alone, they didn't die, versus the one with CAR T cell, they slowly were killed. And we see some different kinetics between these 2 batches. So we think that our platform might be useful for quality control. Towards the end, I briefly want to repeat the key features of the Celldom live cell assays. So every well of the Clone Express 96 well plate has roughly 4,000 microvels, which means that each well contains hundreds of thousands of replicates and controls. And by controls, I'm referring to empty microvels or microvels with 2 cells if you want to study single cells. The cell loading is extremely efficient. Only few cells are needed. We use typically 4,000 cells per well, and all cells end up in microvels. There's no cell loss. We have 96 possible experimental conditions. You can add different cells in different wells or different drugs, peptide treatments. We generate very high, rich data sets based on our high content readouts, time-lapse images of cells. So we have metrics from morphology, cell-cell interactions, cell growth, and cell death. We measure secretion and surface markers. And last but not least, we can recover viable cells after the assay, expand them further, or lyse for sequencing. This is a short reminder of the 4 applications that I talked about: the no-wash fluorospot with optional recovery of selected microvels, TCR antigen library screens with the engineered Chuckert K562 system by Selector, TCR discovery and validation, and the CAR-T potency assay. And with this, I want to thank you for your attention, and I'll be happy together with Alex to take any questions.